So we're a little bit more shallow, I think, than we were previously. We're at 840. We
Yeah.
can go down another 20 meters, or we could run a transect along 840.
We could run a transect along 840. That would be just fine. We're back into a nice dense coral and sponge assemblage here, so I don't think 850 is particularly a magic number.
Right. Does that work for nav and?
Sure, if you just take a look, Steve, you have HIPAC up on channel 3?
Yes.
Yeah, so it looks like the 840 meter contour may take us, it will be less steep, and that's totally fine. Just want to make sure that's what you want to follow.
What do you say, Meredith?
Let me look at that contour real fast.
Sure.
Jumping over. So where are we looking?
So we're down, there's the ship. We're just northeast of the ship here, and we're roughly at the 840 meter contour right now. So that's going to,
yeah.
If we follow 840, it'll kind of be northwest-ish. So it'll be a less steep slope.
Yeah, probably.
I would tend to stick to the steeper slope if that's possible.
Okay.
Although it may have us pointed downhill.
We can get down to a steeper slope and then face the hill and grab along as we were doing before.
Why don't we do that and stay in the steeper habitat? Because I think if we get back into the flat, it'll just be what we just got out of.
Okay, sure. We'll just get set up for that then. Get the vehicles
Thanks for pointing that out, Rennie.
Sure.
So is that, I'm looking at quad view. Is that little mark to the northwest, is that 3C?
Yes,
okay.
Roughly to the northwest of us, or northwest of us, 3C. So the 3C looks to be on a bit of a deeper contour, but I know it's a bit of an arbitrary-ish point, so
and if you want a better look at that, you can always pull up the channel 3, which you can go to full screen with the HIPAC.
There's a nice big pair of gorges here.
Yeah, really big.
Can we fire a Niskin right here?
Sure. I think Argus is stable, so as long as you're happy.
Wow, this is really beautiful.
So we've only been seeing the smaller Paragorgia Linux, and now we're suddenly into this big, this will probably be Paragorgia arborea, or what we've been calling arborea. They're really beautiful.
Maybe we don't need the steep slope to see all the big coral.
So there.
Yeah.
This seems to be...
Sorry, your mic's not on your...
Sorry.
No worries. You get a good shot in Argus there for the scientist on shore to check out.
Oh, yeah.
So...
Zoom in there, Steve, while
Which Paragorgia was this again, Meredith?
On that thing.
Wow.
I think this is what we would call arborea, but they're now not sure that arborea actually occurs on this coast, so it would be either arborea or pacifica. I'm looking quickly at our list.
Zoom back out, I'll pan over to the one on the right. See if I can get it better. No, it's behind the rock.
Yeah, I would call this Paragorgia arborea.
And can we get a couple shots without the lasers on?
Sure, trying to center it up.
Thank you.
How's our DSC on those? Is it looking down over the rock at them? Oh yeah, beautiful. We're getting some high-res shots with our digital stills camera there too. You'll see more of the trunk.
Oh, this would make a great Christmas card. And it's great to see the polyps are out, which is unusual.
Mm-hmm. Feeding in the current.
I can do it. I'll get a good polyp zoom here. Okay, Steve. See if you can fill the screen.
Full zoom.
Really?
Yep.
Quit moving the camera.
Is this an octocoral? Does it have 8 tentacles?
This is an octocoral. In the
Oh, it's so
Paragorgidae.
beautiful. So the reason they call it an octocoral is because the polyp has tentacles, has 8 tentacles. And then I guess on the tentacles are other structures that filter the food out of the water column, the small particles.
Yeah, they can use the tentacles themselves to capture food. All octocorals are in the cnidarians, so they do have the stinging cells, nematocysts, that define that group as well.
Uh-huh. That was awesome.
I'm gonna move a little closer there. I can move up on this. Yeah, we're gonna do the Niskin in a second, but see if I can scoot up here a bit more. Sorry, mushroom.
And
pretty
crab.
resilient.
That crab is scooting out of the way. And what is the coral in the foreground? Next
right
to
at the base?
the stick? The stick?
Do you want to pan right and we can fill with that big wide one?
The light's not on it very well.
Yeah, you're right.
That little stick is just another small Paragorgia. Just hasn't turned into one of these big, beautiful fans yet.
So how old would you say this Paragorgia is?
Ooh, I don't have a good estimate on this species growth rate, but this is another one that can get quite old. At this size, at least 50, probably 100 years old. I would have to do some digging in the literature to get a better idea. I know when we see bamboos of this size, you're usually looking at 200 years or so. And if you're looking at like Primnoa of this size, are usually in the 50 to 75-year-old range.
So very long-lived.
Very long-lived. Paragorgia is one of the groups that can get huge. In New Zealand, they actually have a sample at NIWA that came up in a deep sea trawler in, I think, the 1980s. That is as large around, the base is as large around as a tree trunk. A large tree trunk. Yeah. So this species can be quite long-lived and can get quite large.
So if it's like a tree trunk, that is in the thousands of years old.
Possibly.
So folks, we've been calling this the paragorgids, but they're also common. The common name is bubblegum corals, and no wonder why. Well, it's nice to know that Santa Lucia Bank can also have large corals.
These are actually the biggest corals I've seen in any part of this bank, which is
I was going to say the same thing. Yes, so that's why I said that, because I've seen some some examples of what they found on the express dives or other surveys from MBARI, and I had never seen anything like this. So
really
thank
impressive.
you. Yeah, it is for this area. And we need to thank Kurt Whitmire and Guy Cochran. I was talking with him about where we should go, and they both recommended this area.
Mm-hmm. Guy Cochran is online with us as a scientist ashore right now.
The base of that.
Oh yes, thank you, Guy.
Massive.
Wow.
Got this lovely spiny king crab hanging out here in the branches.
I mean, look how big of a crab it can support.
Yeah.
Mm-hmm.
More off in the distance too.
Trying to get a spot where we're balanced here in the breeze. I don't know if that'll happen or not. A little too close for our DSC. They'll have a really good shot of the...
It's a cool shot in Argus right now. That's with the zoom, so
yeah.
be aware.
Get the size of his tree trunk there.
Oh, nice. That's a great shot in Argus right now.
And you just wanted an Iskand, or do you want to sample this?
I would like a Niskin, although as we move around here a little, if we see a smaller individual, because I don't really want to disturb this big guy, if we see a smaller one of this type, I might sample a piece of it.
Oh, is that one on the rock itself that you said maybe
Yeah, if we could, once we take the Niskin, if we could go back and look at the little one on the rock and see if it's the same, looks like it's the same species. Because we are seeing 2 species here.
Is that all the zoom in, Steve?
Full zoom.
All right. Your porch light on there? Watch your eyes.
This is when we say striking. When you're on set and you're turning a big light on.
Yeah, it might be too far in.
That's a wonderful view of the polyps, though.
Look at those branches.
Mm-hmm.
Sub's drifting a little there.
It's just so dense too.
Yeah, it has such dense branching. All those polyps open.
This is a cool shot. Good idea coming around to the side, Dan.
Yeah, money shot there.
What are those little things crawling around the stalk on the left?
Those look like isopods or small worms, possibly. Oh, they look like little polychaetes. They're kind of crawling around there, little scale worms.
Okay, we'll move back onto the rock there for the Niskin. See that little guy you were talking about? A few more DSCs on the way out, hopefully. You're talking about the little lone guy on the rock there?
Yeah, this taller one, I think might be the same species, or the one to the left that's the darker pink. Well, that might be a branch of a bigger fan. Yeah, this one right in the middle of the grouping that looks like it's got 2 twigs, I think is a good candidate.
Can zoom in on that guy, Steve. Yeah, you can put the lasers on.
So you were saying this is a different species, you think, Meredith?
So this one I think is the same species as these big fans we were looking at to the right, but just to the left and off camera, I think is Paragorgia eulinix, which is like Tom's group will call it the peppermint coral instead of the bubblegum coral because it's got the pink polka dots and it's much whiter.
So maybe after we collect here, we can look at that one.
Sure. So you wanna collect the guy we're looking at now, do you?
Yeah, if we could collect a piece of this one we're looking at now.
Can do. Can you show us the manip there? Thanks. You want the branching bit, do you? Or?
Just that tall portion right at the top. I just need a small, like 2 inch piece would be plenty of material.
Copy. That was a miss. Those coral cutters do come in handy.
Excellent. Yeah, thanks.
Okay, cameras on the starboard box, I'm thinking.
What do you have open over there, Taylor Ann?
Yeah, we got, any of the small boxes are free, unless you wanted to put it in with a sponge. But yeah, you can put it in a small box, any of them.
I think one of the small boxes is probably good for this guy. Yeah. Okay, coming out.
This is quite a diverse little outcrop. We've got what looks like 2 species of paragorgia, bamboo corals, and in the background this white puff that I think is another Peristonella.
Go in box A here too, there.
Alrighty.
Okay, it's in the box.
And that
Is
was
sample
sample.
046?
Yeah. Yep.
Want to trip a Niskin do it?
Yep. Thank you.
So, what's that? Oh yeah, that's all right as long as it. I might do it.
You can go for the second Niskin. It's free.
Niskin 2, copy.
Just switch over to, oh, there you go, you already got it.
Three,
Hmm.
2. Make a challenge for 3, 2, 1. I'm gonna
You
count
are
down.
fired.
I'm gonna count down from 10 next time as soon as you move them, and see if you can make it.
See, I think we're gonna look at this guy right here to the left.
Yep.
Charlie Brown coral here.
Mm-hmm. So you can see this one, the branching is definitely not as dense, much more sparse, and it's a lighter pale color with the polyp, what we call the calyces, the little covering over each polyp are these dark pink dots. So this one I would typically say would be at this depth Paragorgia eutlynix. So it does look like we've got 2 species here. And that's one of the things that the environmental DNA sample we just took will tell us.
Cool.
So for those listening at home, we're taking an environmental DNA. We just took an environmental DNA sample. That was that Niskin bottle that was just triggered. So what I do is they filtered this water aboard the ship, and then they send the filter to me. And what we're doing is collecting DNA directly out of the water. And then back at the lab, I sequence it for all these different octocorals, and it gives me a picture of the local biodiversity and allows us to verify what we're seeing in the image to the species level.
Okay, should we come around and see what else is here?
This is a pretty neat spot.
Mm-hmm.
Yeah, it's right a little peninsula here. Probably get in to have a close look at the other rock down there. Looks like a good spot to get in.
I agree. You saw
Really
something
nice
too,
views.
right, Meredith, sort of
Yep,
in the
it's
background?
just the bottom left corner right now, that white puff. So we'll see it as we move down to look at this other rock. We'll get a nice close view.
Apparently the angle of these rocks is very conducive. It's a big fan. We've both chosen it.
Yeah, they're like duplicate of each other.
Yeah.
Mm.
Mm-hmm. The fans themselves are oriented in a very similar way as well.
Almost looks like they're leaning into the current that we are seeing now.
So do they grow in such a way that they will maximize their feeding ability in the current? I guess that would make sense. So instead of growing towards the sun, they grow towards where the food is.
Yeah, that's how we typically think of them, that you'll see them concentrated and kind of oriented into the optimum feeding position.
What is the white one right there on the right?
In the lower right corner, that I think is Peristinella, which is a primnoid coral.
Look at that crab on
which it is in the octocorals. But a group that instead of having, or its sclerites, instead of being long rods or spindles, they have these flat plates and spines typically.
This is a great shot too with the crab on it, or the crab surrounding it, not just one. There's three of them. There's four. There's one sort of in the background there. So this one seems to have the peppermint dots on it, or not?
I think this one is not. Like, it's also the branch pattern. You can see that this one has that really dense branch structure too. It's not that thinner, more kind of spindly Charlie Brown Christmas tree, I believe Dan said, structure. And so, yeah, when we're looking to do the taxonomy, what we look at are things like the branching pattern, how dense they branch, whether they branch in a bush or a fan, the color of the polyps and the color of the sclerites are also characters. And for those at home, these are octocorals and they're sea fans. And inside their tissues, they have these little sand grains that are all different shapes, and those are the sclerites, and they're what allows them to have this stiff structure in the current.
I'm going to back up just a bit to get we're trying to frame it up for the digital stills camera.
Excellent. And the shape of those sclerites is diagnostic for species in a lot of these.
In the white there, that's a sponge.
That's the one we want.
Yep. Actually, I think most of
Yeah.
these little white clusters all along that lip are all sponges tucked in there.
Oh yeah, it's right down there to the right.
Can I get some without the
And
lasers?
then yes, please.
Lasers coming off.
I'm just going to do a quick zoom in to check my focus here. And I was going to zoom back out real quick. Thanks.
That's really cool.
You don't think
That's
that,
cool.
that's not overexposed there, is it?
That's a Steve question.
On the DSC?
Scroll down, Jake, to the wave.
You can pull it up on your machine over there and play with it if you want while we're sitting here.
Well, if you want to take a few minutes, I
can't.
What's that?
can do it. We got a couple minutes.
Yeah, we do.
We have minutes.
Remember how to get it out with the Control X thing? If you release it there, or you might have to.
Whoa. Just happened there.
Still can. So it's still locked for me.
What's that?
Try refreshing it. The upper right, there's a refresh.
Yep.
Still locked?
Yep, still locked for me.
Right now?
Still locked.
You can unlock. Rennie will help you. We're framed up really nice with the still camera. Steve's been playing with it
It's
a
worth
bit.
waiting.
Yeah, I think so.
Worth waiting to get some good still cam shots.
You can look at it on the right screen there still.
And it's almost as if the crabs are working with us to form a great tableau here.
Mm-hmm.
They've spaced out really well. It goes from top left to bottom right. Isn't that sort of good composition in paintings?
They're perfectly demonstrating why corals are great, what we call biogenic habitat. Oh, there's another crab hanging upside down on one of the lower ones, just in the center of the frame. You can just see the legs poking out from underneath.
Huh.
Really? Oh yeah, we have full control.
Master controller.
We're not control freaks in the front row. Steve's on the case now.
Okay, Steve's messing with
And
the
we're gonna
exposure here to make it nice and pretty high-res photo.
Yeah, that'll be the Christmas card one there. Three crabs to you.
It's actually exposed fine.
Is it? Oh,
yeah.
good.
Yeah, the highlights aren't blowing out. I think it's because these monitors, they just don't have quite the range to show. But yeah, it's all there.
Nice.
Perfect.
I'm not going to touch it. Jake, how do I start this the intervals back up?
It's that button to the right of the record.
Yep.
And you want to move the dialog box out of the way before you start it, or else it gets frozen there.
Copy that, thanks. Does that look all right where I just moved it?
Yeah, that's fine.
Some pause. Interval timer does not start because...
Oh, you'll probably have to make a new folder. I can do it if you want. Unless...
I think I saw where that was. It's a shortcut, right? FTP shortcut?
Yes.
And then...
I don't think you really have to make a new folder, but we've been doing it anyways.
Well, it wouldn't let me
Yeah, it won't let you start.
The,
Oh, really?
Yeah.
Oh, so you do have to make a new folder.
I'm going to call it...
Just add a underscore number to the end of the file.
How about there? And
you go back to the... You go scroll down and change the folder destination. Yeah.
Yeah. Where, let's see.
Go to...
Do you guys want to zoom on anything here, or are you good to move on? Or?
Well, while we're here, why don't we zoom in a little bit on the corals, just straight in, and then pan a little bit to the right so we can look at those small sponges.
Oh, yeah. Okay.
What do you think, Meredith?
That sounds great. You can get a look at the little area tucked right there on the end.
Yeah, since we're here.
Just reading about something over here real quick. Focus.
So, are we recording again? Okay.
I'm just double checking something here.
Right.
Yeah, all this sponge that's to the right looks like Farius. It's a glass sponge. And we've got the start of another little Paragorgia sea fan just springing up out of the crack right there.
Is that what that is to the
yeah.
right? Just kind of sticking out?
Yep.
Mm-hmm.
Surprised it gets what it needs down in there. Testament to the current, I guess.
Yeah. Well, and it's not that big yet, so.
Yeah.
And folks that are listening in, Meredith was describing this white frilly sponge. And if you look at it, you would think that it would be very fragile and soft, but sponges are made up of a tissue that is has like a scaffolding made of glass. So they actually, I was hearing, I think Abigail was telling us that they are more potato chip-like than soft and squishy.
Yep, these ones can be quite crispy and kind of brittle.
Yeah.
And this is one of we have out here on the West Coast.
Do you lose them?
Can you hear me now? I just dropped out for a second.
Oh yeah, you're back.
I dropped out. Okay. I dropped out too. We dropped out at the same time.
Huh.
Solar flare.
Yeah, so these these
the world's ending.
Sponges. We've got 3 types on the West Coast here that when they die, their skeletons will fuse and they basically become rocks And in some places up in Canada, they've formed reefs where there's just layers and layers and layers of these fused glass sponges with new sponges growing over the top.
Okay, we're back up and running on the DSC.
Sweet, thank you.
All right.
Can you
So
put the
in
view up there again?
that way, glass sponges are very similar to reforming habitat in the tropics, I guess.
Yep.
That they leave behind the hard structure upon which successive generations of animals can live and form habitat and more of the reef structure.
So you both got booted off of V-Link at the same time?
It suddenly started doing that thing where it's dropping in and out and in.
Yeah, I only heard the beginning. So you just got booted out?
Yeah, I was asking. I was a little distracted for the last couple of minutes, but I overheard you say that you both got dropped off at the same time.
Yes. And then we, as you were saying that, we got dropped off and then we joined back in at the same time, I think.
Okay.
You want to zoom out there a bit, Steve? And I can.
And does V-Link close when that happens?
No, it sends me back to the login page.
Okay. Yep.
All right.
Okay, thanks for letting me know. We're keeping track of those kind of things.
You can zoom back in. I was just going to pan around. I think they wanted a close-up of the different corals here.
Yes, here's the crab that you saw, Meredith.
Yep.
Holding on.
I can zoom out just a bit.
And pan a little bit to the right.
To the right. Oh, you wanted to see the glass sponge, do you?
Yes.
Copy.
Just that area, because there's another sponge. It isn't just Faria. There's the yellow one underneath it.
Yeah.
Or is that a?
Okay, Steve.
I think that's a demo sponge.
Yep.
Yes, this a little bit more to the right.
Yeah.
Please.
Is that full pan?
Let me.
Okay.
I can.
Oh, you would have to reposition.
I can just wiggle it a little. Should be all right. Hopefully.
A little shake, shake, shake.
Yeah.
Yeah.
Something here.
Yeah.
Some asbestopluma too.
Okay. Should be able to zoom in there
Hey,
now.
Taylor Ann, you're very quiet.
Yeah, I'm a little sleepy back here, but I'm here. Taking notes and stuff too. When we were zoomed on that coral, I was trying to draw it for a little bit too.
Nice.
That falls in there, is it?
That may be one of those tube sponges we were seeing up above.
Right.
Nice little shrimp.
And then what is the very frilly branches that are greenish brown?
So those will be either bryozoans or
Bryozoans or hydroids, I think that's the...
Yes, bryozoans or hydroids.
Yeah. Well, I think we're good here. Is there anything else you want to see here, Meredith?
Nope, I'm good. This has been wonderful.
A nice interlude.
Mm-hmm. We can continue on down slope, down the contour a little deeper.
Downslope, roger. A little deeper. 4 or 5 is what we were going. We only need another 70 or so meters. Bridge nav.
I'm very, very pleased with this. This is pretty awesome to have
Yeah.
found this on Santa Lucia Bank.
75 meters, bearing 245.
These corals found the perfect habitat. These slanted rocky slabs here. It's great.
Overhanging too. It's
Yeah.
funny. They're all overhanging.
Yeah, the largest ones are. They're happily fed.
Found that niche spot.
Flat irons.
223.
245.
245.
75 meters, 245, and then we'll see if that's deep enough, or steep enough, rather.
Yeah, so SPL is kicking me off and on.
Mine as well.
It might just be somebody's using, yeah, like you said, damn solar flares or satellite feed being taken up by something else.
Like the crew streaming movies.
Like that's a separate one.
There's a soccer game on in Europe right now.
Well, it's weird because, you know, the video feeds are uninterrupted.
Yeah.
Yes.
So
the bandwidth for the
something coming the other way, I would guess.
The bandwidth for the SPL is dedicated to, I guess, priority over soccer games.
Yeah.
I've been finding that it tends to be stable for like the first 2 hours, and then somehow like hour 3 is the witching hour where it just starts randomly dropping.
Fighting the current.
Yeah.
It's like you're in a blizzard down there. You got the lasers back on?
Yeah, I can.
Thinking that because I saw a giant rockfish back there.
That, and we're going downhill too, so I'm trying to.
The delay on the Argus screen for switching buttons is like 10 plus seconds.
Rock structure has changed a little bit. Got more of these kind of big bouldery outcrops.
Slabs.
I guess this is what we were looking for. Maybe this is the closest we'll get to bedrock.
There's a really nice Peristanella just on the right-hand side of the screen now, right on the end of one of those big boulders.
Oh yeah, the white one.
Mm-hmm.
You want to look closer at that one, do you?
We can keep moving.
Roger. We're down at, where are we at? Are we have a target depth now? So we're down about 860 now.
Yeah, I think
Too
we were
deep?
just looking for a slope. Is this good enough if you want to lateral along this slope, or did you want to come down a bit further? We do have that kind of, it doesn't, it's at this depth, it kind of flattens out for a bit.
Oh, I see what you're
As far
doing.
as transect goes.
We'll keep going down.
Yeah.
Yeah, I'd say go down maybe one more.
Yeah.
That sounds good to me.
Need to get out on the down slope side of Argus anyways, don't it?
Yeah, Argus is facing 135 right now.
No, that's right. Is that right? Yeah.
Argus is 135 if it faces.
Yeah, he's facing
more like
almost up the hill.
045. That'll be up the hill.
She should do when we start going to the north, right?
Yeah.
Stuck on a rock there when it.
Did you see your new
There's a
button
good rock.
labels, Jake?
Yes, I did. Thank you.
That was my day today.
I only knew
Couple of
dive
thorny
and
heads.
sample.
Mm-hmm.
Nice. Now you can, yeah.
Now I know them all.
And that, you know, Mountain ROV3 is the one right in front of you there.
All tucked in.
Are you still getting booted out to the login screen?
Yep.
Yes.
Let me try something. Yeah, just let me know if it when it happens. I'm trying to take notes here so we can get the issues addressed.
It's happening pretty much every minute now.
Okay.
Oof.
For the past like 15 minutes or so, it seems like.
No, for the past.
Oh.
Yep.
Like it just happened.
Yep, I heard that.
Yep, just happened to me too.
Some Pharea. Looks like there's a skate egg
Auto iris.
casing in there.
Sorry.
Yeah.
My auto iris.
Taking notes over here.
Spoiled, Steve.
I can click auto.
But there's another sponge on top of there above the Pharea.
Yep. One of these kind of plate-like sponges.
Uh-huh. But again, here's sort of that slab that is sticking out.
Yeah, the big overhanging lip again. Oh, and a rock just below with all these pits in it.
Wow. What a sponge that's now a rock.
Yeah, sorry,
that's a certain. Yeah, that's just. We should. I'm gonna make note of this and talk to our geologists because that's some geological formation. I don't think an animal did that.
Yeah, it's very interesting. It almost looks like solution holes.
Mm-hmm.
The ship is stopped going down slope. Argus probably has a bit more to go and may yank you off backwards, but we're okay for now.
Just looking for a rock to pick up. I don't see any little ones. All very massive.
Yeah.
Yeah.
Hmm.
Another one of these heterocony sponges we sampled right at the beginning of the shift.
Hmm.
With a crab living in it or something.
Yeah. I think it's a little crab.
Has its nice little home.
Okay, where do I need to be now? I need to be more down.
You're doing great. I mean, we're not on transects. I like sort of just moseying along and seeing what we find.
Yeah.
Trying to work out where I need to be in relation to Argus before we start moving.
Yeah, Argus, if you keep changing your head around to 45, you'll be facing the slope, I think. See it in the sonar as well.
Yeah.
Not a lot of fish here.
No, we're deeper than the range for most rockfish. You'll occasionally get them wandering down here, but they're usually above about 600 meters, other than the thorny heads. We are seeing, I've seen a few snailfish, some eel pout, but yeah, it's not a really high fish density area.
Yeah, like no grenadiers. Wouldn't we have seen grenadiers, or would they be deeper?
You can see grenadiers at this depth. I'm wondering if it's something with the current, if the fish that might otherwise be around are kind of sheltered, so they're not spending energy in the current.
Yeah. So when you see another snailfish, point it out, because it would be nice to check one out.
I will do that.
Thanks.
All right, so I'll start driving a long contour here so we can follow Hercules as it goes lateral here. That's all right. Bridge nav. Let's do 100 meters, 310. Thank you.
You can come down 5 meters or so, 3 or 4 meters.
Roger.
So for folks that are listening in, one of the reasons we are investigating Santa Lucia Bank is this is an area that has been proposed for as a national marine sanctuary. It was nominated by the Northern Chumash Tribal Council, and the name they gave it is the Chumash Heritage National Marine Sanctuary. And the nomination aims to recognize